Hello? Yes. Good. Distance. So hi, I'm Cesar Reno Costa. Okay. I'll be presenting our work with the robots that I'm demonstrating there in the exposition room and with the posters. This work has been done together with Andrea Lovisotto, who is here, and also Alberto Marti, and under supervision of Paul. So let's go. So the first thing I'll talk about the motivation about this work. Well, it's not very common to have a catering word in a Congress title, but we did a lot of work with mobile robots. So my main expertise is basically on models of behavior of rats, especially in special tasks. And we were invited to make a robot demonstration during a fair of the future and emerging technologies in Warsaw, and we had to demonstrate the robot. But my experience that demonstrating models of place cells, grid cells, and for the general public can be not so interactive. So we started, okay, how can we enhance and use our models in the sense to make it more visible, to be able to interact with people. So this was the area that we had to use the robot. It was a big room from the Warsaw Technological University. And we came up with one idea that was solving a catering problem. So basically, what's catering problem? It's just to distribute food, distribute chocolates to the people in the room. That's the point of view of engineering, how can we define it? So the idea is to minimize the demand of food of people. So the room will be full of people wanting chocolates, wanting something, and the robots just had to give them. There are some particular things on this task, mainly that the robot has to be able to navigate, in the sense, has to go to the person and all of them to get the chocolates. But the main thing that's completely different from other models that we've been working with is that the robot has to interact with humans. It's quite different from a model robot, like a humanoid robot, that you can work with speech and verbal communication when you have a robot that it's a tank. So actually, how can you make a tank communicate with a person and give him chocolate? So this was our main problem. And this is the robot that we had. So it's really a big tank. And the input we have is basically proximity sensors, infrared and ultrasound proximity sensors. We had a pen tilt unit with a camera that we can make some gaze control. We have a control CPU, and we have a position to place chocolates. So how would be the classical approach to this kind of problem? So especially in artificial intelligence or any kind of computer-based intelligence. So you normally would define which are the problems of the engineering, which are the environmental variables. Is this working right? So then it would mean that you have to predefine representation. So define what's a guest, what's the room, so what's the self-representation of the robot. Also, you have predefined actions, so which is which is gaze the guest, or which is approach someone or navigate. You would have predefined states, like, okay, I'm close to someone, I'm not close to someone. So this leads to the classical programming that we always see in robotics, that is the state machine. So normally when you're in programming the interface of a robot with a human, there are several of these programs that you just establish. Oh, in this state, I'm close to someone, so now I go to there. I'm not close to someone, then I go to there. There are some problems on this kind of approach that you're modeling the complexity of the interaction. So you're basically, if you come to a situation that it's not predicted by your model, then you fail, basically. And by modeling the complexity, you start generating complexity models. It's a very well-known problem with state machines that the states can just explode. And so the kind of work we try to do in our lab is what we would call, for this conference, more like the living machine approach. So that instead of trying to work with complexity, we're trying to work with the term of the simplicity. Simplicity is the connection between simplicity and complexity, in the sense that a complex process can be controlled or can be ruled by simple rules, by the same way that simple rules can generate very complex behavior. So our plan is not to model the whole complexity of the problem, but instead design for an emergence. This basically means to find which small rules or small, very minimal control rules that we can make so that we can generate complex behavior and solve the task. So using our tools, we already had some experience in the back, especially Paul with interacting with humans in the ADA machine, that we had a big room that we could control and make communication with the users. And also, we play with the idea that the synergy between how the robot is acting with the environment is also modulated by the environment. So the complexity of the interaction is generated also by the environment and not only by the rules you have in the robot. This is more or less the base of architecture that we've been developing for many years in our lab. That is the distributed adaptive control. That's an architecture that's based on the theories of conditioning. Conditioning is this idea that in animals you can have you can have the learning that you associate one signal to another. Basically, that you have a signal that triggers an action, triggers a reflex, and you can learn that another signal that happens at the same time is also associated with this reflex. So the idea of the bottom-up approach is that representations and also the definition of the problem will emerge from initial rules that you have in the system, and not to start from the complete definition of the rule. So how decades are organized, it's divided into 3 different principles of how the cognition can emerge in this kind of system. The first rule is the reactive, that you need to have to start with a repertoire of different reactions, different sensings, and different actions. That's basically what we call, what in this context would be the sensory motor reflexes. So basically you have one sensing that will trigger directly one action. The next step, you would have what's the adaptive part, that you can start to make association between different native sensors, native sensing or native actions. And from that starts to build representation, start to build complex perception and complex action. At the end, at the higher level, you have what we call the contextual learning rule, contextual part, in which you can have this kind of punctual representations and starts to learn relations between them, and relations in the sense of sequencing, in the sense of time, and in the sense of space. More or less the way this architecture is shown for simple robots would be more or less like this. In the part of reactive control, you would have some sensing that is connected directly to some action. So you have, for instance, if you can program a robot to, whenever it detects a kind of light, it's aversive, so it just goes to another direction. At an adaptive control part, you can start to have some kind of different learning on that. So imagine every time you get some aversive learning or aversive to some input or some stimuli, you also receive a red light. So whenever you see this red light, you also get the robot to learn that you have to be aversive to that situation. And then this kind of thing can trigger different reactions or a conditional reaction. At the contextual layer, what you start to do is that this kind of learning, this kind of associations between a sensing and an action, you can make sequences out of them. And this kind of sequences create an organization. This kind of organization, depending when you are whenever you have a goal or you have a kind of signal that causes that it's relevant for the system, then you store it to your memory. The thing is that the action of the robot then can be controlled in 3 different manners. Can be controlled by a direct sensing that you're receiving, can be controlled by an adaptive sensing that is a direct link between 2 inputs, or can be controlled by a contextual control that depends on how your history of acting and sensing. Well, under this kind of framework, we did a lot of different works on traditional, more robotics kind of task. For example, this is a sequencing task that the robot had to learn which are the colors it should look. So basically, it will gather the information and make a decision. So whenever, if it's the wrong decision, then it's get a red signal. Whenever it's got the correct decision, then it gets 2 other sequences. That it has to learn the full sequence at the end of the game. The thing that we just start a system with basic actions like go forward, look for a color, or something like this. And at the end, it will, by learning, whenever it gets the goals, it can have the correct behavior of actually deciding which are the correct colors. Another part of this work, for example, is the idea of creating the representation of the position only based on getting the information from the sensor of the robot. So here we work with models of grid cells and place cells based only on the information of the wheels of the robot. So we can build a representation of the space based on that. And another kind of experiment that we worked on this framework was also the emergence of spatial learning and spatial behavior, in which the robot had to associate to different positions a place where he can find food or where he can find other things. So basically we had like a grid on the space that we could emerge the representation of position, and then he receives a cue, a visual cue whenever he's at his home, and by exploring the space he can associate a cue to a certain location. The thing that everything is emerging from initial set of rules that with time it can exhibit the expected behavior. But the thing about these tasks and the catering task is that all of these problems, they have a very well-defined goal, a very well-defined behavior that we were expecting from the robot. And that's not the case of catering. Because catering, the idea is that, well, the new constraint you have is that now we needed to have human interaction. And human interaction in the sense that we cannot quantify it exactly if it's good, if it's not good. So our question here was, how complex do we actually need to allow an effective interaction? An effective interaction by means that someone sees the robot and feels the will to go there and catch chocolate. So what we proposed as a minimal solution was just the development of a reactive layer to solve the task. Reactive layer in the sense to have a system that is non-adaptive, so there's no learning involved, and that this kind of system can base as an adaptive behavior system. Can, in the future, by having a minimal behavior, we can, based on that, to learn and optimize what would be a good behavior of the rat. The thing about the human-robot interaction is that it can very easily look fake when you're in a robot. Fake in the sense that if the interaction is extremely reactive, like the robot always responds in the same way to the same problem, then it will just look very artificial in this sense. So the idea that we were working on this paper was basically to have an internal drive regulation of this kind of sensory motor reflex. So just a sensory motor reflex is just a direct link between a sensor and an actuator. So a sensor sensing the world will just cause like a motor response or like this. And the drive is just a way that you could regulate the actuators and the sensors. But this drive is also only defined by the activity of the sensors and the actuator. The idea of how you can create complex behavior out of this is just that the drive can work in a different time scale than the sensory motor reflex. That's because the sensory motor reflex is instantaneous. So it's always like in action and reaction. So with a drive, you can modulate how is this action and this reaction and give the sense that something more complex is happening. So, and what kind of different drives you could have? So basically, you can have some linked to homeostatic needs, for instance. For example, if the robot is hungry or not, this will make the reaction to seeing food different. And also, it's important that it's set dynamically by the system itself, so that it will evolve in time and give different reactions. And the most important thing is that it follows a different time scale than the reaction, than the sensory motor reflex itself. So the principles that we designed for the robot are basically that for navigation, it's just moving around. So you just have to move around in the sense that it will explore the whole environment. For the communication with the guest, we follow two types of non-verbal communication. One of them is gaze. So just we try to make some complex gaze behavior that the robot tries to look to faces and establish some kind of connection. And also the idea of the smooth reduction of interpersonal distance. That's just the idea that if the robot is approaching someone and changes the speed, slowly changes the speeds close to someone, gives the idea that it's approaching and starting some kind of communication channel. To implement that, we implemented two control loops, one for the gaze, one for controlling the vision, and another for controlling the loop, the locomotion. So we have like the visual loop and the navigation loop. And this is more or less how the system is put. So the idea is really like, how can we be minimal and still have some kind of complex behavior for interaction? So for the visual loop, we have the action is the control of the pan tilt of the camera, so it's the gaze of the robot, and the perception is the information from the camera after some visual processing. So more or less what happened here is that Action can be caused by some random spontaneous activity, so you can be gazing wherever you want in a random fashion, or it can be controlled by the perception, or it can be controlled by the camera. What happens that the camera is always winning the competition if it's competing only for the random. So the function of the drive is that the drive is modulated by the action. In the sense, every action will minimize the drive, but the drive also has some incremental spontaneous activity. So the idea is that whenever the robot gets stared at too many times at a person, or stared too many times to some position, it will start to inhibit the perception. So this loop would cause that the gaze will never get stuck at some person. It will always be varying between looking at someone or looking to random positions. So the idea of how is the gaze implemented is basic that we have a higher bank of filters to detect faces. So from the detection of face, we can create a salient map of the visual field. And from this salient map, we can map into different reflexes of action. So depending on where in the visual field, the camera will basically try to take the most salient point into the center of the area, so establishing a gaze procedure. So this is basically more or less what happened, that whenever it detects a certain face, it will gaze to it. So this is just a video of it at work with different parts of the navigation, and how the gaze also works with the robot moving. This is the area that we had to make the demonstration. So like, this is the robot with the gaze control. Just an idea how it's the vision that you have from the robot. So it had some sensors that just to avoid hitting people and this kind of thing. And this is just showing more or less which is the behavior when it's navigating. In this case, I'm controlling it, so the navigation part, it's bypassed by the controller. So just the visual loop is working. So basically the idea is that you can, that we could see a lot of reactions, just like you're not expecting it, just you see a robot coming by and starting to stare at you. And this is more or less the kind of communication we want. Also, the navigation loop followed more or less the same idea of just having a drive managing the perception and the action loop. But in this case, action was the control of the wheels, and perception was the function of the proximity sensors. The difference in that in this case, the drive was only linked to action, because perception was just a security measure to ensure that it was not hitting at someone, especially a big robot like that. And so what was happening that the action was a random input for moving, so like it could go forward or it could turn. And the drive was just causing a different cycle that would cause the movement to be at certain points to reduce and stop, and at certain points accelerate. The idea is just that at many times it would stop, and it would be the time that the person would have to go and collect the chocolate. And the kind of this kind of very simple algorithm, the thing that it covers the space. Like we could make some simulations on different size of rooms with a virtual version of the robot, and show that like here the graph you can see that this is the trajectory of the robot, and this is the point where it stops. So the idea is that it can cover more or less uniformly the space and distribute at certain manners to several persons in the space during the catering job. The thing that we modeled this for the size of the arena that we needed was this more like 4,000 square meters. So like in about 1 hour, it could have like 60% of the whole arena. It could be covered just by the robot. So basically, the thing is that after 10 hours of demonstration, we could deliver 213 candies. But the thing is that it was sufficient to cause some kind of interaction, in the sense that people could go there, could get the chocolate, and can make some kind of interaction with the robot. But that's definitely not optimal, in the sense that it can get stuck at several parts of the environment, like can spend too many time in a region that there's no one, everything. So the thing is, this kind of reactive and extremely minimal system can behave as a good base for this kind of task, or any kind of human interaction task. But you still, to have something that can do the job, you still need some enhancements. So like that's a kind of work we are planning for. It's like to have some hippocampal-like navigation, so the robot can emerge the learning that I've been here before, so I already delivered chocolates in this region. You can have some face recognition in the sense I already gave chocolate to this one, so he doesn't want some more, so I can go to other places. And also, this would involve memory in the sense I know him, I already gave to him, and try to find some new people. That's basically it. This is the references. So I mean this is the scheme of, I think it's the third version of DAC. So like this is not exactly how it's the scheme that we implemented on this version of the robot. But if we would compare, I was working only on this part of the scheme. So this scheme is just like an architecture that shows how you can emerge some kind of cognitive or some immersion cognition on this robot on different tasks. So the details here is normally that you always start with a set of links, a set of sensors, a set of actions that you can work with the robot. So for example, in the case of the sequencing, we implemented some actions that were just like go forward, look right, look left, and this kind of thing. With this, we had an implementation in we have a neural system that we developed in our lab called IQR. So it was kind of neuromorphic relationship we would have there. And for that case, then you would have to start in the adaptive, when you start, for example, you have sensors for colors, for different colors, like we have basically the hue information. And then you can start to associate the hue with looking right, the hue with looking left. It's a neural network or it's a symbolic? It's a neural network, Because here, basically, the association is created by this part, is internal states that you have, that it's basically a big matrix that is making the relation between the whole space of the CS and the whole space of the US. So you're doing some Hebbian learning that you're making the connection between these two parts. Yeah.
Sorry.
The thing here is that in this part this connection is direct. So whenever you have an input, you're directly causing an output. And in the way the system is like here, you can only modulate this by another stimulus. What the difference here is that this modulation we are doing some internal drive in the sense that it's not modulated by any external cue. So the direct connection between the sensation and the action is regulated internally with some different time scales.
Any other question? I have just a small remarks. I was wondering whether you can, the robot can use its movement while it is interacting with the person to understand what the person wants. Because I was, for example, in the video that you show, the person was moving backward. Mm-hmm. So probably somebody that wants to take a chocolate will stay in place, and the robot we can recognize by the fact that it's not moving.
And how these minimal versions, the two loops are completely independent. So like it's always gazing, and it's also always moving, stopping, moving, and stopping. So the idea that you would need some kind of learning to actually make the relation between these.
That you might add later on.
Yeah, but at the moment, no.
Then a follow-up, and maybe this is something we can also discuss later in more detail, but there's always this issue about benchmarks, right?
Yeah, definitely.
And the different kinds of benchmarks in the field of robotics. So do you think, now with your experience with this catering robot, do you really see this as a benchmark that we should try to establish that every year we have a competition? I mean, as a benchmark, what does it add compared to other benchmarks that you would know? So do you really believe, would you really push this as a benchmark for, let's say, living machines of this kind? Or do you think it was an interesting experiment, but as a benchmark it's just not sufficient?
Well, as a benchmark it's quite hard because it's not so easy to quantify, especially because you have to give a lot of chocolates to get it statistically relevant. And also you have to compare to, for example, if you just put a table there and people are getting chocolate, or you have the robot moving around, and then make some comparison, this kind of thing. So it's not something very practical to use as a benchmark for robotics. But also it's a very human interaction in this sense, and social interaction. It's also very hard in the sense of benchmarking. So I mean, it's definitely an interesting paradigm, but as a benchmark it's quite hard.
Okay, I think we have to stop here, and if the next speaker can come to set up.
thank you. Okay, that's good.
So the next talk, as you see, will be high-resolution biohybrid device for probing the brain, and will be presented by
Stefano.
Thanks.
Welcome. So, I'm actually from the biohybrid community, not from the biomimetic community. So I really hope to say something interesting for you, also because I and we think that the two communities are quite well linked each other, and so I hope that after my talk we can have some little discussion about that. So. The main message is just one or two slides. Is that if you want, many of you are interested to mimic the brain, or at least some functionality of the brain. And of course, if you want to mimic the brain or some functionality of the brain, you have to first understand how the brain works. I mean, this is the usual preferred pathway, let's say. And this is a tough job. It is a tough job because the brain is a complex system, of course, and you need not only structural information, which is absolutely relevant, but also as much as possible functional information, and you have to relate structure to function. And this is essentially a technical problem. How to get good functional information from the brain. In order to do that, you need to establish essentially an efficient communication mean with the brain. And we will see now what we believe is efficient communication. Particularly, you have to be able to interact with the building blocks of the brain that are essentially, not only of course, but the main building blocks are neurons and neuronal circuits. And establishing an efficient interaction means reading outputs from the brain and from the neurons at high resolution, and also providing inputs in order to probe your hypothesis. And this means to establish a bidirectional interfacing with the brain. The problem, of course, is that the brain is, and even much simpler nervous systems like those of insects are 3D structures, and particularly the human brain, of course, and you must establish a good communication not only with the cortex, but also with many other structures, even deep structures in the brain, and that are interacting each other, that are all contributing to perform processing during behavior. So this leads essentially to the technical problem. Of course, we have, on one side, we have tools that can get access to the neurons in a very high spatial resolution, with very high spatial resolution, like the usual intracellular recording techniques. High spatial resolution, I mean that you can get not only single neurons, but you can really measure from different compartments of the neurons if you want, and analyze how processing occurs at the individual neuron level. You can also, they have also high sensitivity. I mean, you can record action potentials, which are, as you well know, the real bits of information in the nervous system, but you can also get access to much finer details like postsynaptic potentials, subthreshold oscillations, and so on, which are also relevant for single neuron processing. However, this has a price, because if you want to use this kind of technique, as you know very well, you cannot get access to many neurons at the same time, which is unfortunately what is needed if you want to study brain function. as a let's say in terms of behavioral tasks. And also another problem is that if you want to study behavioral tasks, you need a stable connection with these neurons. And through these techniques, intracellular recording techniques particularly, is extremely difficult to get stable intracellular recording for a long time. On the other side, you have, of course, a wonderful toolbox of techniques for large-scale recording. I mean, techniques that are allowing you to study many neurons simultaneously, whole brain areas like functional magnetic resonance imaging, electroencephalography, and so on. But, as you know very well, they are not, they do not provide a high spatial resolution. You measure essentially from populations of neurons, large populations of neurons, and you do not reach a high spatial resolution detail. Another problem that you have to cope with is that they do not provide high sensitivity. In which sense? In the sense that you do not measure individual action potentials, but you generally measure global neuronal activity in terms of fields, in terms of metabolism modifications, and so on, but definitely not individual bits of information provided by neurons. So the question that, of course, the driving force for developing new tools, new interfaces for getting in touch with the brain is this one. So we need techniques that are capable to record from individual neurons and from many neurons at a time with the resolution, with the sensitivity sufficient at least to measure individual action potentials or some small population neuronal activities. Stimulation is also a problem. I mean, as I said, you want to record, but you also want to provide inputs to your system. And it's in a specular way, you have the same problem. Either very high resolution techniques for stimulating individual neurons, or very rough techniques allowing to interact with the brain, non-invasive techniques that can be used during behavior, but they are large scale, like transcranial magnetic stimulation, for example. You provide big stimulations to large portions of the brain, but it is despite the importance of these techniques, it has not a great value if you want to understand how brain works at the microcircuits level. Okay, given this premise, the basic idea, which is very old, I mean, it's not definitely new, is that if you can put in your brain some sensors with respect to recording or actuator with respect to stimulation that are capable to record simultaneously from different neurons, you can somehow understand how a neuronal microcircuit in the brain works. This is a typical case of a tetrode. This is a very old technique. Only 4 electrodes are placed in this case in a cortical column, and by using triangulation techniques, you can more or less understand which neuron is firing at which time. This has, however, a limitation, which is here depicted. This is a paper that I suggest you to read if you are interested, by Buziaki. You can get access to a limited number of neurons in this way, because you have only 4 electrodes, and you can more or less, by rough estimation, you can more or less record from neurons that are located 50 micrometers away from your probe, and you can measure or identify spikes that are generated by tens of neurons and not much more. Okay, so this is why, since from tens of years, there was a strong push to develop new techniques, new recording and stimulation techniques that are called multi-site recording and stimulation techniques in order to have a large number of electrodes to be placed in contact with the brain. This is a very known and typical example. They are so-called Michigan probes. They are like shank electrodes. Each one is featuring several electrodes. Maybe in this picture is better visualized. You see each shank provides 1, 2, 3, 4, 8 electrodes. They are spaced, I think, 100 to 100 micrometers apart. And so you increase in this way the number of available electrodes for recording. And again, by triangulation techniques, correlation algorithms, and so on, you can record from a larger neuronal population. Of course, what you aim for, if you want really to study, as I said previously, the brain, is to study neuronal processing during behavioral tasks. This is the best way. You do not want to study the brain while it's silent, is sleeping, okay, but you want really to study it while it's doing some processing work. So the problem is to have tools that can establish a real stable and long-term connection with your neurons. And this means, to my opinion, a biohybrid device. The best and more antique example is the use of microwires. This was introduced by Nicolelis several years ago. And through these bundles of stainless steel microwires, you can get beautiful long-term recording implants. Of course, each one of these microwires is recording only from one side, so you have bundles. And in each bundle, you have one record. You have wires that are recorded individually from one point. And here you see in the monkey how you can realize multiple implants to probe different brain areas, even in depth. This has, unfortunately, a resolution which is not really very high because the bundles are, the single microwires are separated by hundreds of micrometers. Stimulation works also through microwires, and recently it was coming back to biohybrid, really biohybrid systems. Yeah? It was possible, also in the group of Nicolelis, to establish a bidirectional interface. So the microwires were used both from recording from the motor cortex and for stimulating the somatosensory cortex, so that in this case the monkey was fed back also with the monkey was navigating in a virtual environment, moving with the joystick a virtual arm, yeah, and was provided with artificial sensory feedback in the somatosensory cortex to get some, to try to feed the monkey with some sort of sensory effect, yeah. And this proved to work, at least in terms of velocity of learning tasks. This was then a stable implant, a stable interface was also achieved in patients. You probably know already the work. I mean, but it was in the so-called Utah probe. These are 100 needles probe, which was implanted in a patient in the motor cortex, and for recording from the motor cortex, and the patient was capable through this interface to move an artificial arm or a cursor on the PC. Something like you can get, however, also with the EEG interface, by the way. This is more or less the state of the art. So in this case, in this talk, in the remaining part, I want to describe you the concept we put forward in this direction to obtain a stable bidirectional large-scale interface with the neurons. So the idea is essentially to increase even further the number of electrodes and in our probe, in our interface. So large scale, even larger scale with respect to the state of the art. Higher resolution, because at the moment the previous probes I've shown you have a resolution of several tens of micrometers in the best case. We wanted to go down to about 10 micrometers, and also having a real stable probe and for behavioral recording and stimulation. So bidirectional and stable. So as you see here, the concept is that we wanted to explore at least the possibility to interface in different ways, either by implantation in the cortex, or in deep structures as well, or by putting the probe on the surface of the brain to record cortical activity with a high resolution electrocorticogram. What I mean for high resolution, you can have an idea. This is a work that was developed also further during the project by Peter Frommerz and colleagues at the Max Planck Institute. Here you have such a probe. This is a 128 times 128 sensor array. This is designed for in vitro experiments, but however, you can get a feeling of what you can achieve in terms of resolution. If you put a piece of brain, so a slice on the top of it, and you record from in this case, an hippocampus slice, you get something like that. Now, I hope that the video works. Yes? No? So I was prepared to that. Let me just 1 second open it separately. Okay. So you stimulate, there is a stimulation from a conventional stimulating electrode, the emulsifiers bundle, and then you have a propagation of the electrical activity of neurons in the hippocampus. And for the more experts, let's say these are field potentials, local field potentials that are, can be detected with a resolution of about 7.8 micrometers. So you can do a real electrical imaging of your piece of brain on the chip with a very high resolution. This is more or less what we want, what the aim is to achieve such a high resolution also in vivo in a behaving animal. Of course, these are some certain constraints, but at least this is the driving force, let's say, of our work. Okay, so in principle, with the same approach, instead of local field potentials, you can record also from individual neurons with also some tricks, however, either directly or using correlation algorithms. And this is an example of individual Purkinje cells recorded from a slice using the same chip I've shown you before. So high resolution, large scale, this is was our aim. Another difference with respect to available probe is that we aim for really stable interfacing. And the current probes I've shown you are based on metals. And this has some problems. Yeah? This raises some problems. Yeah? So particularly, as you know probably very well, when you place a metal in an electrolyte and you apply voltages, you have some redox reaction. This is not too bad if you record. I mean, the currents passing through the electrode are quite small. But this becomes a problem where you want to stimulate, this potentially becomes a problem when you want to stimulate, and possibly in a physiological mean, in a physiological way. When you apply large voltages for stimulation, you pass big currents, you have redox reactions, you have modifications of the chemical environment, and this can, for example, pH can change, and this can affect neurons. So the idea is, very briefly, so this is how these probes are actually the silicon-based probes. You have microelectrodes in contact with the electrolyte. The idea, which stems from Peter Frommer's actually, is to have an interface, an electrical interface, but through an insulating layer of silicon oxide and more recently titanium oxide, so that you do not have Faradic currents, you do not have chemical reactions, but you do have capacitive currents. So only ions are displaced in principle. So using a transistor structure, for example, you can record voltages in the tissue. Let's assume here there are neurons, okay, they generate voltages, you can record them. Or in a different configuration, you can stimulate. If you use a purely capacitive structure, you can apply voltages and stimulate by capacitive currents neurons in the tissue. We of course aimed for proof of principle experiments to see whether this approach could work and could work also in animal, of course. And these are the two prototypes that first were developed. You see here, these are silicon needles. This is the tip, featuring either transistors, 4 transistors, this is the first prototype, or capacitors. These lighter areas are the areas where the silicon or titanium oxide is thinner in order to allow for capacitive coupling with the tissue. This is actually how the needles are more or less looking like at a lower magnification. You see the sensors are here. Now in the latest designs and prototypes, we have up to 256 recording sites, and we now aimed for 1,000. And this is the part of the chip which features the contacts for the readout electronics. And this is the mounted probe. You see the chip is mounted on a PCB, and this can be connected to a PC and implanted in the animal. So these are the two basic configurations that you can use to test your probe. So the first one is in the anesthetized animal. You just implant you just open the skull and you implant your chips. In this case, it's a multiple implant. One of the first experiments we did, motor cortex and somatosensory cortex. Or later, we also tested chronic implantation, of course, and this is how the implant look like in the living, freely behaving animal. Of course, here there is, as you will see in the video in a moment, there is a cable coming out here for connection to the PC. So we tested the system in the living animal after, of course, a lot of testing work also in vitro, and we took as a benchwork the somatosensory whisker system of the rat. Maybe several of you are already familiar with that. Just very briefly summarizing, the whisker is provided with a receptor which which fires and feeds information to the trigeminal ganglion. The trigeminal ganglion then projects to the thalamus, and then from the thalamus you get a nice even at the level of the thalamus, but particularly in the somatosensory cortex, the so-called barrel cortex, you have a very nice region where columns are present, so-called barrels, and each barrel is actually receiving information from one specific whisker. This is actually very, very broadly speaking the system, so that when you deflect artificially a whisker, you can have a response in a specific barrel. So the idea, of course, at the beginning was to test whether the recording with these transistors works at high resolution, and so one of the more recent proof of principle experiments was done with a 2 columns probe. So 50 transistors spaced 40 micrometers. We have different topologies, even 10 micrometers, but so 40 micrometers pitch and 2 columns inserted vertically along the barrel profile. This is one of these barrels receiving inputs from one whisker. And this is something that you can get. So these are actually field potentials recorded from the 2 columns of transistors and along all the through the cortex, yeah, from the upper layer down to the bottom layer with the resolution of 40 micrometers. With a different representation, you can get perhaps a better feeling. This is the time domain, you see. Here, there is the whisker stimulation, and here, after a certain latency, depending on the depth where you are in the cortex, you record a different response in terms of field potential in this case. You can go even down with the resolution. This is a 10 micrometers profile, and this is actually the highest resolution that has ever been reached in such a kind of multi-site recordings in the brain. We are now working, of course, on taking advantage of this technology for recording single spikes. This is a population activity, but we want to record also single spikes now. Finally, well, of course, we tested also for histological damage. One of the drawbacks of this, potential drawbacks of this approach is that the size of the probe is bigger, about 300 micrometers wide and 100 micrometers thick. So with respect to the Michigan probe, this is, of course, much worse. But our thought was, okay, if people are anyway taking out slices of the brain and they provide anyway interesting information and conserve neurophysiological properties, I mean, even if we introduce a bigger probe, this perhaps should not harm so much and should not impair completely recording capabilities, recording the signals that we want to record. And indeed, the histology confirmed that this damage is not too high. Finally, to complete my talk, I wanted to show instead the other side of the medal, so the stimulation. So is this approach of stimulation through capacitors working? So this is a first evidence that is working. So we provide a stimulation with a capacitor in the somatosensory cortex, and we record the response in terms of local field potentials with the second probe. So there are stimulation with one probe and recording with another probe, and you see that you have a response indeed in terms of local field potential. But what tells you really that the thing is working, again, is behavior. So is, at the end, this approach working the living animal? And the answer seems to be positive. Okay. No, sorry. This is the other one. Okay, this one. Okay, sorry. So the configuration of this experiment is that the animal is implanted with 2 chips. And each chip is in the either left or right barrel cortex. And the stimulus through the left or right chip is provided by pressing the left or right button. And then also the animal is provided with a third electrode, which goes to this MFB, which is the medial forebrain bundle, which is a bundle in the brain which is, when stimulated, providing a highly rewarding sensation to the animal. It's called the pleasure center of the animal. And so this is used for a highly effective rewarding stimulus. So when the animal enters the T maze, here the animal receives either the left or the right stimulus. And if the animal performs correctly, he gets the reward. And so you train the animal, you condition the animal to respond to the stimulus. And indeed, after a few trials, about 30 trials, the animal learns to respond correctly to stimulation, and with 100% efficiency. So this means that really stimulation works through these probes. And this works for several months, at least. Then the animal was killed for histology, but it's very stable. And finally, the other evidence that you can also somehow tune this stimulation and provide different degree of stimulation. It can be seen here, where you increase the intensity of the stimulus. You shouldn't be scared by the voltage, because this is a capacitive stimulation. So by increasing the voltage, what happens? You see that when the animal receives the stimulus, it throws back the head, it has a twitch, and this is increasing depending on the intensity of the stimulus. Like the animal is feeling something, maybe an object, because it is stimulated in the barrel cortex, and then is retracting the head, and then is going ahead. And this is actually providing evidence that you can modulate indeed also through this kind of approach, stimulation. So, and again, with this lasts over months. So since my time is over, apparently, we did also some work on the interfacing from the cortex, but I think I will just provide with the conclusion. So essentially, at this present stage, we can say that the recording and stimulation capabilities are proven. Of course, as I said, we are aiming for a single spike recording now, which is the other side of the medal. We want to provide both capabilities, local field potential and spikes. One interesting feature is that these chips are compatible apparently with functional magnetic resonance imaging. So you can somehow arrange to do experiments, combine experiments with the technique. And then for future work, I mean, one thing we would like to do, besides other things, is also to provide these chips for optogenetic stimulation, of course. This is a must. Okay, so these are the members who contributed to the work, and thank you for the attention.
One question, and possibly the next speaker might start to set up in the meantime.
Hi, thanks for the nice talk. So I was interested, I mean, the critical point in any of these technologies is going to be the sort of spatial resolution that you can both activate in terms of, populations of neurons, and also separately detect with, say, spike sorting or other multi-unit detection. So this is going to be very crucial to any level of bandwidth that you can affect between an artificial and biological system. So I was very interested in this point.
Yeah,
so
indeed. Thanks for the comment. Yeah, indeed, There is some work ongoing, and actually some work that has been done on algorithms especially designed for spike sorting using the high resolution of these probes. And this is one of the potentials. Okay. Thanks.
So what sort of numbers of neurons? What sort of numbers of neurons do you think you can say? What sort of numbers of neurons do you think you might be able to say stimulate and separately record?
Okay, so for stimulation, we still have the problem that in order to get enough current, we need to use relatively large capacitors. So anyway, at present we stimulate populations of neurons, probably tens of neurons, by eye. It's not really, it has to be investigated. For recording, this is a tough theoretical question. I mean, of course, with such high number of sensors in principle, you can record from probably maybe hundreds or thousands, probably, something like that. It depends on the interface, on the quality of the interface, and so work has to be done, of course. Thank you.
Okay, thank you again. So we are moving to the last presentation for this morning, that is going to be presented by Navid Eyaz, I suppose. And the title is Static versus Adaptive Gain Control Strategy for Visual Motor Stabilization.
Yeah, thanks very much. Well, after the previous talk, I feel quite humbled when I'm now talking about an interface using just one electrode. But anyway, let's see what it can... Yeah. The talk is a little bit misleading. I had to rearrange to connect better to the general topic of the conference, biomimetics. So I will try to give you a general idea of what I think biology can bring to engineering, and engineering can bring to biology in a way. Now, the title could be, if I get this, yeah, multisensory motor control, actually. What I put together here is actually a number of photographs I got from Roland Hengstenberg once, and I added a few by myself, and they make a very good point in terms of general principles of motor control in biology. So if you look at all these different examples of humans riding motorcycles, birds flying, and then this guy here playing some sports, famous figure by the way, or a cheetah hunting, no matter how complex the control task is, you can find some common feature in terms of the overall behavior. And that is if you just look at the head in all these different examples. And I play the little video again, which shows the fly in the center of it, which does basically the same. So when the thorax is changing in its overall orientation, the head is immediately compensating for that. And it turns out really that this sort of compensatory gaze stabilization or head movements, that they are quite instrumental in a few respects, one of which would be to reduce motion blur. The visual system wants to analyze, so to speak, all the visual information around, and to be able to do that in a more efficient way, it is stabilizing the gaze. Now, another reason for most of these animals actually is to keep the head aligned with the inertial vector. Because all the head-based sensor systems, like in humans, the vestibular system, in flies, some other systems I will be talking about a little bit later, which are head-mounted, they all then share the common frame of reference if they compute anything related to self-motion, which is important in terms of overall control. Now, this is what we discussed briefly yesterday as well. Now, flies and jet fighters, what do they have in common? Well, certainly not the price, but they have in common that both are extremely maneuverable, and it was mentioned yesterday already what the secret is behind this maneuverability. It's aerodynamic instability, because if you're totally unstable aerodynamically, then you can, as fast as possible, basically change your momentum or change your state altogether. So that is quite a common feature between the two of those. So if you throw them in the air without any feedback control mechanisms in place, they will both crash right away without any chance to recover. There is no passive aerodynamic stability in build. Now, flies use vision a lot. Jet fighters don't use vision, well, not really vision, not in that sense. But maybe it is nice to look at the different sensor systems. I will do that later on as well. But let's stay with vision for the time being. It tells you something about the design principles of sensors in biology, and also the control strategies connected to the constraints of sensors in biology. So in biological sensors, you have normally very noisy signals, constraints in terms of bandwidth, all sorts of limitations which are due to the bare fact that biology is dealing with fat proteins and water rather than silicon. Now, of course, in silicon circuits, there are also limitations. We'll get back to that pretty much later. But so how does biology mitigate these noise problems? And the secret to this is massive integration. So all the sensor systems we know of, at least in insects, consists of a huge number of individual sensors, all of which are noisy. But if you integrate the signals appropriately for a particular task to extract certain parameters which are important for control, then you win. And that's what biology seems to do. The strategy is then sensory integration, multi-sensory integration. That means different sensor systems are used, each of which, within each of which there was integration taking place to start with. And that is a massively or fundamentally different strategy from these guys. So they take only a few measurements to learn about the state changes, throw these measurements into 3 supercomputers, and the output of those supercomputers is then issuing some activation commands which then work on the control surfaces. So there are fundamentally different principles. And of course it would be interesting to see whether we can exploit some of the principles found in flies to control some micro-air vehicles or robotics in general. Right, okay. Vision. I stay with vision here, but just to make a statement on how successful it might actually be if we look into very small nervous systems as opposed to the human brain as we saw before, or a vertebrate brain. The big advantage in the nervous system of insects is we know a great deal about the general anatomy. This is, by the way, a horizontal section through the head, showing the different parts of the visual system. Here, the retina, a couple of neuropils. And then here is a flat structure, LP labeled, which is called lobular plate. And we will stay with that structure because it contains a number of neurons. And actually, per part of the brain, left and right part of the brain, it's only 50 to 60 neurons, which we know individually because we can stain them intracellularly and then see how they look like. And they are looking the same from animal to animal, really. And also we can then do intracellular recordings from them and look at the individual, the voltage changes in these neurons depending on visual stimulation. Now, the point why I bring this up is really that we have identified neurons here. And we know in where they are situated exactly and what they connect to. So these neurons output to the motor center, which is controlling the flight motor, and also to the motor centers that are controlling the gaze at the same time. Right, now let me give you an idea of how we can actually study the way how these neurons respond to visual input. And I didn't mention yet, what they are particularly selective to is motion vision. So it's in which direction is a stimulus moving when the cell becomes active. Now, here's a little cartoon, which I hope you will be able to appreciate. You will see in a second a little stimulus that is moving, and at the same time, while the stimulus is moving, the activity of one of these neurons I showed you before is recorded with using I hope you could hear the correlation between the motion of the dot and then the periodic responses there. So you could just see that during a certain, can play this again, during, it's a little bit, during a certain motion phase, there's a massive increase of the spike rate, and if the dot is moving in the opposite direction, then there's an inhibition. So you can use these responses and analyze, well, actually you can apply, first of all, you can apply this stimulus to many different positions throughout the visual field of an animal. And I didn't mention before, flies have 4-pi vision, so they can see everywhere, except for a tiny little bit that is obstructed by the body. Many of these neurons have only a visual field that comprises one visual hemisphere, but some have visual fields or receptive fields, actually, the neurons, which combine both parts of the visual field. Now, the data are then very simply, or in a straightforward way, they can be analyzed. This is, in fact, an intracellular recording, but basically it's the same principle as we saw here. So the dot is moving, you get a sinusoidal modulation of the membrane potential. You chop it into individual pieces. You can then, or can run it through a Fourier analysis, whatever you want. And then you can extract 2 parameters. One is, what is the phase angle where the response is maximum? So that gives you the so-called preferred direction of the neuron. And then you can also extract another parameter that is basically the modulation depth at that particular location. Now, armed with these 2 parameters, you can actually start plotting little vector fields. And this is actually done for 3 different individually identified neurons here, the so-called vertical system, 1, 6, and 8 neurons. And if you do apply the stimulus within really one entire visual hemisphere, then you find these receptive field organizations, which are quite characteristic. And all those of you who have been working on optic flow processing would immediately see that this is an optic flow field, or very similar to an optic flow field, that's generated when the animal is rotating around the roll axis. Now, this one here has a singularity about at 45 degrees. That means there is an intermediate axis between a pitch rotation and a roll rotation. So this is an additional axis for something also sensing horizontal rotations. And this one here, the VS1, would be best excited if the animal is performing a nose-up pitch. Now, the other system I mentioned a little bit earlier is the horizontal system. I showed that before. You can do the same analysis for all these neurons as well, and then you come up with something like this, which is very sensitive to a yaw rotation. Now, it's just the vertical rotation axis of the animal, and then if it's turning around this axis, then this neuron would be maximally active. These 2 together, if you combine all this information, then it is actually quite good also to sense forward translation. Right, so from these receptive fields, you can actually use an algorithm that was proposed by Kundering and Van Doorn and extract the motion parameters these individual neurons would be tuned to. And that's what we did a couple of years ago to just see what sort of preferred rotation axes the VS system or the VS neurons are covering. And what you can see here is a rear top view on basically an animal or the body axis of the animal, which would be sitting in the center. And this is the roll axis, so the longitudinal axis, that is covered by VS6. I mentioned that earlier. Then here, these 2, VS1 and VS2, would be sensitive to pitch rotations, and the other neurons are covering axes in between. And this is really interesting. So it's not a Cartesian coordinate system in that sense. So we have a massive overrepresentation of rotations in space just if we look at pitch and roll, because all the there's a number of intermediate axes which are covered there as well. Right, now how far, or what can we do with this information already? Now, a couple of years ago, Sean Humpert and I met on a conference, and I realized that what Sean had done in the meantime was just to look at these axes, take these axes, put it in a linear control engineering framework, and then use it for quadrupter control. And it's very straightforward. You can just, if you know the vehicle dynamics, so that's very clear, you can for quadrupters anyway, and you have the quadrupter moving in space, then and you have some optical system that can catch the optic flow, and then the optic flow is projected into all these individual receptive fields as a template or a matched filter, so to speak, and use the output. Then you can just feed it in your weighting matrix K and use that as immediate feedback on your vehicle dynamics. And it's very straightforward to do that. He has done that in this. You cannot hear it. Lucky you. This is a nasty noise. So what he uses it for in terms of control parameters is just orientation of the quadrota, forward speed, and distance to the walls. The ground distance is still conventionally controlled by distance measurements. But it's no problem using optic flow. For that, that's work from Marseille, for instance. That you can use optic flow as well to control altitude in a very straightforward way. Now, this is not really what I would consider highly maneuverable. It works. The proof of concept is fine. You can control some parameters here, but it is not what the fly is capable of. Now, the fly is not only using vision. The fly is, in fact, using a couple of sense organs, and some of you might have heard of them. So we have, obviously, the compound eyes. That's what's responsible for analyzing motion vision, and that's what I described so far. And then we have another system, which is a mechanosensory system called the halteres. These are former wings. So in four-winged flying insects, they were the wings. And later on, these comparatively young flies, the so-called dipteran flies, it was transformed into some sort of gyroscope-like organ. So it's beating up and down in antiphase to the wings, and it has a couple of mechanoreceptor fields down here at the bases. Now, if you imagine you have a beating system that is attached to the thorax, and the thorax is undergoing a sudden change. Because of the inertia in the system, you will have strain exerted on the structures of these halteres. And that sort of strain, and actually the temporal pattern of that strain, is measured by the halteres and can be used for flight control. Now, the good thing, and you all know that probably about the mechanosensory systems in biology, they are extremely fast, while the visual system is comparatively slow. And that's actually nicely demonstrated in a behavioral context here. This is actually on the y-axis, always plotting the gaze stabilization behavior. You saw that in my very first slide, against the angular velocity of the stimulus. Now, if we look at a pattern that's been moved around the fly, with the fly being stationary, and we know it's only the compound eyes and motion vision that's contributing, you get a peak in the response of the animal at around 100 degrees per second. Now, if you then have a homogeneous visual environment where there is no visual information to be had by the fly, and you move very quickly the fly and rotate the fly very quickly, then the peak that's actually picked up by this system would be at 1,000 degrees per second. Now, if you look at the two dynamic ranges here, they complement each other. And yet, we have to keep in mind that they have different time delays in the processing, which, if this is not carefully adjusted, can kill feedback control. That's why, actually, the mechanosensory system very often is used in a forward way, while the remaining disturbances are then picked up by the visual system, which is then brought up in its operating range, and then it can be used in a negative feedback way on the same parameter, say gaze stabilization, or also flight control. Now, there are other mechanisms, for instance, these so-called ocelli. I think they were involved yesterday already in some of the talks, which actually sense changes in light intensity. So you've got 3 expanded light sensors, which have huge acceptance angles, and by checking the the differential illumination between those, you can actually tell whether you are rolling or whether you are pitching. That's what a fly certainly does. And that is an interesting system which is way faster than the compound eyes or the motion vision pathway. And this is not only because the pathway itself is so short, but it is also because processing just light intensities takes less time than processing direction of motion. Direction of motion is only to be had if you have a delayed, well, a certain mechanism that requires more time to work out what the actual direction is. So this is actually a system that speeds up all the sensory motor responses altogether. Now the big question obviously is, how are these systems all interacting together to do, say, gaze stabilization or flight control? And well, Roland Hengstenberg, who did most of the work here in gaze stabilization anyway, he suggested that it might be just an internally scaled signal coming from all the different modalities, added up, and that's it. It is not that easy, unfortunately, even though I would like that. It is in many cases that there are some nonlinear processes. Well, it is on the other hand, it is very likely that it is happening that all the systems are combined somewhere. And this is actually one of my favorite figures, which is certainly not one of the favorite figures of the students I show it to, because it looks rather complex. But the only point I would like to make here is we have one center in the brain, in the so-called lateral protocerebrum, that integrates information coming from the ocelli, coming from these VS cells or HS cells I mentioned before, so the motion vision pathway, and there is ascending information bringing signals from the halt hairs here also into this area, and from there again motor neurons are activating the neck motor system, descending neurons are then going back to the thorax and then activating or actually controlling the flight motor. So it's all, we know the pathways in a way quite well. And from some experiments on neurons like this so-called V1 neuron, which is one of those tangential cells, here's the overall receptive field of that neuron. Actually this time it's binocular, so it can see everywhere basically, and it's tuned to sort of bank turn movements. In this neuron, and it is a spiking neuron, so you could look at the action potentials, it was very obvious that if you would stimulate the ocelli, you could introduce extra activity. So this multisensory integration can be seen already at the level of these particular neurons. And that is quite interesting. So the trace down here is a raster plot where from 0 to 1, there's just spontaneous activity, so completely random. And then when Matthew Parsons, who did the experiments, is alternatingly stimulating the ocelli, mimicking some sort of rolling motion, whatever, you can see that there is a phase lock response in that neuron. And this is supposed to be the motion vision pathway. So another example, and that was actually a study by Stephen Houston a while ago, who looked into the receptive fields and properties of neck motor neurons controlling the gaze in the animal. He found some classes of neurons which would actually only accept or would start spiking if the halt hairs and the visual system would be activated. And if they would be stimulated only by the visual system or by the halt hairs, there would be no spiking. So this is the intracellular recording. During this period of time here, there is a motion stimulus presented to the neuron, and you can see it's just a subtle depolarization here of a couple of millivolts. But then down here, if during the same time you move the halt hairs up and down at a frequency either at 100, I think in this case it's the lower frequency, I'm not entirely sure, so you can do it at 120 or 100 hertz, it doesn't matter. You get always some postsynaptic potentials in the neuron, but no action potentials. Only if you present both at the same time, then you get the full-blown action potentials, which is necessary to make the muscles connected contract. So there is some multisensory integration on, and it's difficult, though, to get hold of it and understand how in greater detail this is actually managed by the nervous system. So there is another problem with all these experiments. Of course, all experiments have problems. And it was mentioned in the last talk as well. What you want, ideally, is to record the activity in the nervous system while the animal is behaving. Well, we saw some really nice data from the rat where you can do that using these techniques, multi-electrode recordings and so forth. But what do you do with these small animals like insects? So that's one problem. The other problem is you want to stimulate all the different modalities in a way you perfectly well control, ideally. Well, that's also quite tricky. So at some point I thought maybe it's a good idea to approach this problem by just taking a fly and putting the fly on a robot, and then move the fly around, making sure that the dynamics of the robot still work in a range, so I can just stimulate the halteres as well and can stimulate the visual system as well, and then see what I get. Why would I imagine that I get maybe different results in terms of electrophysiology as opposed to the experiments I'm presenting here? Well, we have also heard about the forward models biology is quite frequently making use of. That means it's a prediction about the sensory feedback you receive given you perform a certain action. And this is what in some systems has been nicely demonstrated at the neuronal level already. Electric fish, for instance, is one, or there was some work in crickets, for instance. And I'm quite convinced that forward models are the answer to many tricky questions in understanding biological systems. Anyway, so we used to set up such a brain-machine interface. We picked a particular neuron that is sensitive to horizontal motion because we stay in 2 dimensions. Okay, so I see that I will have to go through this a little bit more quickly. The first step towards having a fully integrated system where the fly is actually controlled by the robot is just showing an existence proof that you can set up such a closed loop system with the fly, and the fly is in this case still not integrated on the robot, it's just sitting there in front of computer monitors. But the input the fly gets here is a robot that's sitting on a turntable that is rotated and is seeing a stripe pattern. So obviously there's some image slip, and the image slip is picked up by high-speed cameras here, which move then the pattern on the monitor. We then record the activity in the fly and use that activity. We transform this into a control signal for the wheels of the robot and basically compensate for the externally induced motion. I can see that. Okay, so it's a very simple system, Azuro, equipped with some communication Bluetooth channel. The images have still to be transmitted by cable because that would be exceeding the bandwidth of what we have. Okay, I'll just show you what you get in this case. I think this is a constant motion of the turntable. You can see that here, yes. And in this case, it's just a proportional controller, so there's not very much to it, and you can see that it drifts. But intermittently, it's actually catching up with the motion that's induced, and it's then compensating for it. So this is a very simple approach, a simple proportional controller. I'm afraid I will. So I'll just. Okay, so I go to a last. Well, the structures are actually in the abstract. I'm very sorry for that. This is how the adaptive controller works. Now, where do we want to go with this? This is a characterization of the controller. Where want we go with this? Now, this is slightly different. This is actually a platform that will go on the robot in the end, where the fly is mounted in the middle. And I don't know if you can hear it. No, you can't. So it's. You can see here a recording one of those neurons, those spiking neurons, and the whole point here is that the PhD student of mine could actually set up a system here mechanically with everything miniaturized, from the amplifiers to the micro-manipulators to hold the electrodes, and it's all integrated on that aluminium-based top for a robot. It's a slightly faster robot as well we want to use for that. And actually, it is very robust in terms of the recording. That's the point here. So you can really do whatever you want, and you will have a stable recording. This is the next step then. And then finally, what we are working on at the moment is a micro recording probe of 1.1, a floor plan of 1.1 millimeter, low power, low noise system with 4 channels we can use. This is actually a slightly different layout of the whole thing, which we have succeeded to build, and it is actually as good as a 1.8 K high-spec extracellular amplifier, so it's working really well. The only thing we are missing are the electrodes. So we have, because of the scale we are talking about, we really have to have an extra chip with electrodes that can be connected then and make it a functional unit that in the end goes into the head capsule of the fly, and then using radio transmission as a module or dumping the data on die-like flash drives, we would just record while the fly is moving around. Of course, we will have to record it at the same time, but that's the ultimate goal of the project. Okay, so I think I don't have to make that point with what benefits you get using modern and new technology to investigate into the brain or into a system, and then spinning off wherever you can technology that can be applied to different areas as well. My thanks are in particular to Navid, who couldn't make it, Navid Eyas, who is actually an author of this paper, and Raiko Tanaka, and then Manos Drakakis, who is an electrical engineer in bioengineering, Sean Humpert, who implemented the control system using optic flow to control it, and then Chris Patterson, who did the micro-recording probe. Okay, thanks very much, and sorry for overrunning.
And we have a poster and exhibition section now. I think the speaker can take the question during the break, during the poster section. And so we can stop the section here and start to go to the poster, and then we'll reconvene at 3 p.m. as usual. Thank you.